And RNA-seq data analysis. Who have heard about RNA-seq before? You should have, right? So can you please briefly remind me what is the main difference in transcription between prokaryotes and eukaryotes? Really briefly description. Brief description.
Splicing.
Splicing. What else? Okay, so we need to finish up some biology, right? So, transcription in bacteria, what do we have there? We have a promoter region that is typically located downstream. We have a gene region that is located upstream, gene coding region. And it's typically a continuous gene coding region, so it's not separated. It's going just in one line. We have a transcription starting site, and we have a transcription terminating site, with a terminator inside, which I found quite a funny term to go with. So we have also a cis-regulatory region that is located typically upstream over there, and this cis-regulatory region is regulating the transcription of our gene, right? So this is what's happening in bacteria, and in bacteria we typically have a circular genome. However, in eukaryotes, transcription is much, much more complicated. So what do we have? We have two DNA strands, and we have introns and exons, where exons is our coding region and introns is our non-coding region. And transcription regulation is much more complicated again than in comparison to bacteria, because in bacteria you typically have really nearby cis-regulatory region over there, and in eukaryotes the regulatory region can be located nearby, but it can be also located thousands of base pair away. So if it's located nearby, we call it cis transcription regulation, and if it's located really far away in another chromosome, for example, it's called trans regulator. So when we read DNA by polymerase, we generate so-called pre-mRNA, premature mRNA. And to remind you how many RNA transcripts we have, how many types of RNA transcripts we have in a cell. We have a lot of different types of RNAs, right? We have tRNAs, we have rRNAs, we have long non-coding RNAs, we have message RNAs. And today we will be talking mostly about message RNAs due to the lack of time, and because RNA-seq is most of the time used for mRNAs actually. So when we have our premature mRNAs, we still have our introns inserted to it, right? So those introns should be cut out when we generate our mature mRNA. And when this mature RNA is generated, we call it splicing. So we cut out introns, and then we get our mRNA transcript, continuous RNA transcript. And this is used as a template for protein production, right? And it's not necessarily that this mRNA actually result in a protein product, but it may be. It may be also degraded in a way to protein production. So again, the transcription regulators can be located really nearby, but it can be located elsewhere, thousands of base pairs away. And the regulation in eukaryotes is very complicated. It typically involves a complex machinery of proteins that regulate this production of RNA transcripts simultaneously. And again, we have introns, we have exons, which we need to splice. And so what we have in eukaryotes, we have alternative splicing. So when you're splicing your introns, you may splice it in a different way. You may cut out one exon in one isoform, let's say, and you may leave this exon in another isoform. And it may result in different protein products. So you may have different protein isoforms at the end. Why do we need to remember about it when we're doing our RNA-seq analysis? Because we will see those isoforms in our RNA-seq, and we need to be able to recognize those, right? So why do we sequence RNA? We can sequence on different levels. You've already heard that we can sequence on the DNA level, we can sequence on the RNA level, we can sequence on the protein level. And we can sequence different type of information. We can sequence also methylation site, we can sequence where we can actually check where polymerase is located on a DNA on a specific moment. And we can also check where this specific protein is located in a cell. Those are different type of information, and different type of technologies are used to obtain this information. But the underlying law stays the same. DNA, RNA, protein. It never changes, right? So why do we sequence RNA? What do we want to do with it? What is our main purpose of RNA sequencing? Can you name at least one? Function. More? Okay, so the first question that we can ask is, what is our RNA that is expressed in the cell? We may want to address a specific RNA transcript. Let's say that we know that RNA transcript is a signal for some specific disease, so we may be interested in a specific RNA transcript or a specific pathway to sequence RNA. The second reason is if we would like to see whether some of the transcripts are differentially expressed in different conditions. Let's say if we mutate a gene in one cell and we keep a wild type and we compare RNA transcripts after mutation and in the wild type, and we'll see whether there is any difference. So what is the effect of this mutation? We can also treat with different drugs or the same drug two cell lines and see whether these drugs have effects on our cell lines and whether this effect is the same or actually different. So we call it differential expression analysis, and you have seen it probably already in the other courses too. And the third reason why we want to sequence RNA is to obtain a specific feature, so to do actually fundamental study, like what are the different isoforms of a specific RNA, so to find alternative RNAs. The second can be fusion transcripts, so sometimes we see that in a disease state some genes are actually fused together, and I'll come back to it in a moment. And we would like to see those fusion transcripts because it may be important to analyze them for cancer, for example. Or we can also study RNA editing, something that is happening after transcription. So fusion genes. Fusion genes are found to be important in cancer. They are the results of chromosomal aberrations events. So we can have deletions, insertion, different kind of processes happening in the cell. Again, cancer is a disease of the DNA, right? So you might have chromosomal rearrangements that leads to different mRNA protections. So let's say that one chromosome arm is copied somehow to another chromosome arm, and we might have two genes fused. So in healthy cell, they are normal genes. In a disease cell, they're fused together, those pre-mRNA transcripts fused together in mature RNA. And with RNA-seq technology, you might actually be able to see those type of events. And this is called end-pair short reads for example, Illumina sequencing, and you probably also have heard about it previously. I will come back to end-pair short reads later on. And this is one of the examples of such a fused transcript that you might want to study with NGS, with Illumina sequencing. This is a mouthful name, TMPRSS2, ARG. It has been found in prostate cancer in 70% of the cases. So what it's doing is disrupting AR signaling and leads to invasive carcinoma at the end. It's a complicated process. The result is invasive carcinoma. So if it's found in 70% of the patients, it would be really important to actually analyze this and maybe try to generate a drug that is able to address this fused gene. Actually, the results of the many studies that has been done on this gene are quite controversial, and there are no drugs so far that address both transcripts at the same time, both genes that are fused. As far as I remember, there are drugs that address only ERG, but it's not specific, and it may actually ruin healthy cells too, so it's toxic for healthy cells. So this is the example why it's important to study RNA, and so why we can use this information to treat some diseases. It's a slide describing how RNA-seq is working, how the technology is working itself. So we have our mature mRNA transcripts, and if you try to sequence all mRNAs located in the cell currently, most of the time you see that it's actually rRNAs that is over-exceeding the amount of mRNAs. So you need to deplete rRNAs first. And you can do it in different ways, and one of the ways is actually select mature mRNA transcript using poly-A selection. So you add oligomers with TTT sequence, and you select your mature mRNAs on the basis of poly-A tail. When the selection is done, then we need to fragment your RNAs, because we are still not able to, well, with newer technology we're able to sequence alone regions, but with current technologies that we're talking about, RNA-seq, we're not able to sequence very long regions. We're able to sequence only a couple of hundreds of base pairs, right? So we need actually to fragment our RNA first, and then we generate cDNA, complementary DNA, with the help of reverse transcriptase. So we generate our cDNA because it's more stable than RNA, and we know how to handle it a bit better. And then we generate, we prime the generated strand and do a second strand of cDNA synthesis. At the end, we finalize by addition of adapters that are complementary to adapters located on a slide that is generated by Illumina, right? So on the slide, we have adapters, so we hybridize our generated cDNA sequences to the slide, and then we start our PCR amplification. So when PCR amplification is done, we get our sequencing. And it's happening similar to I described in the first lecture. So we wash out with different nucleotides that are marked with fluorescent labels, and then we check the light intensity for different fluorescent labels on each step. We wash out with the first nucleotide, we check what is the light intensity. We wash out the second one, we check again. So we do it multiple times, depending on our required sequence length. Let's say it's a couple of hundreds base pairs, and then we stop and wash out first strand. In the paired end read, we also generate the same, we also repeat the sequencing for the reverse strand. And this helps us to identify actually two reads from the same strand. One is in this direction, another is in the reverse direction. And we know, more or less, we know for sure what is the distance between those two. So this is transcript 1. And this is transcript 1, 2. Why it's helpful, I'll explain a bit later. So we have 3 types of alignment. After you generate your DNA sequence, and after you sequence, you get your sequence in the file, and then what do you need to do? What do you typically have? You have a read that is mapped to one transcript, right? Or it can be mapped to many transcripts, actually. But you need to align all of those reads that you are obtaining from RNA-seq. And you can align against the transcriptome with known isoforms, and you can align against all exons in genome, or you can actually first assemble your reads and then align. So why would you use 3 different approaches? What is the benefit to use either of those? So for example, why would you use alignment against transcriptome? When is it important? No ideas?
It's the knowledge we already have.
Sorry?
Oh, because we're doing RNA-seq, we're looking for a transcriptome.
Yeah.
Comparing it to what you already know.
Yes. So it's the fastest way, actually, right? Because you have some preliminary information, you align your reads against existing and known isoforms, but this is a biased way. So you say, okay, I think that this is what I should get, all of the known isoforms, but actually you might lose a lot of information in this step. And you are doing this in case you're just interested in any gene that is aligning to a specific region in the DNA. So you're not really interested in a specific isoform, but you're interested if this read aligns against this gene. Why would you want to align against genome? Why would you want to align against genome?
As it says, you lose, well, while going from DNA to RNA, you lose the exon
Yes.
and then to enable to get into
So with all exons, you're also getting information about splicing events too, right? Well, at least you're aiming to do that. So with genome alignment against genome, you are less biased. And you can try to identify novel isoforms, which you cannot do with the first approach. And why would you want to assemble your reads first and then align? What is the purpose of this third step? So if you don't have known DNA transcript, if it's not a model organism that is not well characterized, you might want to align, you might want to assemble your reads first in a de novo approach and then to align it against known genome to identify what are the transcripts in this less characterized, less well-known organism, right? And when this approach doesn't work, for which sequences it won't work? If you have low complexity sequences with lots of repeats, we are not able to align it properly just because we don't know where this read is coming from which piece it's coming from. And when you have your RNA saccharides aligned against genome, again, you might be able to identify different splicing isoforms. And this is where paired end reads comes into play. So we have our single end reads, and you have your reads that is mapped against your genome. And on genome level, if you have a read that is coming from one edge of exon to another edge, like this piece actually is missing in your RNA reads, right? You don't see it in your RNA. And if your aligner, if your software that you're using or that you're writing is actually not taking this into account, that you don't have this information on your reads, then you might lose these reads completely. And that is decreasing your coverage. So you're less covering your transcript information. You're losing some information. If you're aligning on a transcriptome level, then you are better served because you have these exons connected to each other, then you don't lose this information about introns. But what about these two isoforms, isoform 1 and isoform 2? Let's say you have reads that is coming out from exon 5 in isoform 1, and another read is coming from isoform 2, the same exon. Would you be able actually to recognize from which isoform it's coming? Probably not, because those are the same sequences, right? So you can try to estimate it using some statistical metrics. If you know the relative abundance of isoform 1 and isoform 2 in your sample, you might estimate whether this read is actually coming from isoform 2 or isoform 1. And for paired end reads, you have two reads coming from one side and from the other side, so you know that those pieces are connected for sure. And again, if you're mapping against the genome, and if your aligner doesn't take into account the intronic regions, then you might be in trouble because you're again losing this information. And if you're mapping against transcriptome, you are not losing this information anymore. And why is it helpful for fusion genes? So if your genes are fused, and if it's coming from completely different regions in chromosomes, let's say from two different chromosomes even, with paired end reads, you will be quite sure that those two exons are connected, or those two exons are connected, because you see those two pieces together, right? And then you might say, okay, this actually, the exon that is coming from gene 1, and this exon is known that is coming from gene 2. Probably then it's a fused gene, actually. You might have different technical errors coming from PCR amplification, for example, that will generate those type of events, even if it's not really happening in a cell. But there are different also types of correction for these type of events. So the idea behind paired end reads is that you are able to identify, first of all, different isoforms, and you are able to identify probably fused genes. So yeah, again, why would you align against the genome is because you are able to identify new transcripts, you are able to identify fused genes and more splice variants. Also, because you don't know actually where this transcript is coming from either from 1 or from either from 2, you need actually more coverage as well to be more sure which read is coming from where. When you have more coverage, you have more intersecting sequences, more intersecting reads, and then you are more sure in when is this event actually happening and where this event is actually happening. So, as I mentioned before, there are different types of aligners that takes into account the centronic region, so they break reads. If it doesn't map to anything, they break reads, and then see whether it's mapping to different regions and exonic regions. So they take into account the splicing, and those splice aligners are top hat and map splice. And why would you align against transcriptome? It's actually much, much faster because you allow it to be less exact, and it's not unbiased anymore. So with alignment against the transcriptome, you are not able to identify novel transcripts and splicing options. But it's fast, and if you don't need to be exact, then you're better to go with the transcriptome level. Those unspliced aligners are BWA, bow tie, and shrimp. I don't know if they're actually the most used aligners. So imagine that you have two genes, gene A and gene B, and they are homologs. So they share some sequence. This piece in gene A and this piece in gene B are the same. And imagine you have two conditions, control condition and treatment condition. So you have your reads coming from gene A, you think that they're coming from gene A. You have reads on gene B, and again you think that they're coming from gene B. And then treatment condition, you see that probably gene A is overexpressed because more reads are coming from gene A. And you see that those reads are actually mapped, can be mapped to gene B as well. Then how would you separate? How would you tell if gene A is you had library size 600, and the second case it's 1200, and it's for the same transcript in sample 1 and in sample 2. So again, would you say that in sample 2 actual transcript 1 is more abundant than in sample 1? No, right? So you need to correct for the library size and you need to correct for the transcript length. How would you do it? What do we do to correct this type of things? Yeah? Yes. So a metric that is called RPTM, not a metric but normalization that is called RPTM, reads per kilobase per million mapped reads. So what does it do? We have 10 in the power of 3 that is coming from the kilobase. We have 10 in the power of 6 that is coming from million, because we typically count our reads in millions. And then we have our actual number that we counted from the RNA-seq technology, and we divide it to the library size and to the length of the gene. That is how we normalize. So each value that is coming from RNA-seq, you normalize for the library size and for the length of the transcript. And this works well for single-end reads, but we have also fragments per kilobase per million for paired end reads, because you have paired ends and you cannot apply the same metric anymore. So those methods were quite early, but now we use something else. We have DSIG package and we have HR package that uses different type of normalization, and I will show a bit later why exactly we switched from this type of normalization to another type of normalization. So again, why do we sequence RNA? We're also interested in a differential expression, right? Between different type of treatments, or between wild type and a mutant, or between a diseased individual and a healthy individual. And because we're analyzing expression data, we need to also understand what kind of statistics we want to apply to this. Like if this gene has this amount of information on it, if this gene has this amount of information on it in a sample to you, how would you compare them? You need a statistical measurement for it, right? You need a statistical test. So you can imagine mention that those reads are not full in normal distribution. You don't have minus values in it, right? What kind of distribution you would expect it to follow? If it's counts, there is famous statistical distribution from statistics, yeah, that is quite known for counts. That is Poisson. So you might want to use Poisson, but you also have non-parametric test or permutation test. For a non-parametric test for gene expression data, you might want to use Wilson-Rankson test. What does it do? It ranks your genes, it ranks your reads in transcript 1 and the same transcript 1 in sample 2, and then it compares them and generates your p-value without implying any kind of distribution. But yeah, and there are also permutation tests. What are the permutation tests? You shuffle the labels in your samples, and then you try to see how many false positives you get if you repeat the same analysis many times, how many false results you are getting out of it. And then you might say, okay, if I'm getting false results, false positive results, just by shuffling the labels, it means that in my real results, when I'm doing a real data analysis, I might have this amount of false positives. So it might help you to estimate the amount of false positives in your sample. But those tests are not quite powerful. And typically, to make it more powerful, to identify slightly, events with a less effect, you might want to use some other tools like lemma or string bias. As far as I remember, both of them are using, I apply the Bayesian approach, and you might want to use HR and DSeq, which imply, both of them imply distribution, but not Poisson one. So Poisson distribution is good for modeling counts, and what Poisson distribution describes is a probability of given number of events happening at a specific fixed interval of time with a specific rate. The intuition behind it is imagine an emergency room, and you know that in half an hour you might expect one patient. So this is lambda equals 1. One patient is coming every half an hour. And you might want to ask yourself, okay, what is the probability that I will see 5 patients in this half an hour? And the probability if your lambda equals 1, this probability is very, very low, right? But if your lambda equals 4, so 4 patients in half an hour, then probability to see 5 patients is still low, but much better than 0, right? So this is how you can reason about it. This is the given number of events, probability of a given number of events in a fixed interval of time with a rate lambda. So why Poisson is a good model? Because, yeah, it's just models our given number of events in a fixed interval of time. And if technical replicates exist, then Poisson is a nice model. But if you introduce biological replicates, which have more variability, then Poisson distribution found to be not as effective as you might expect. So why it's not effective? Because it underestimates variation. In biological replicates, you will have much more variation than in technical replicates, and you might want to add this up to your model. And typically, the current software packages using actually negative binomial distribution. Negative binomial distribution introduces an extra parameter that is called dispersion. And this dispersion parameter helps you to catch this variation that is found in biological replicates. So the formula is a bit more complicated than the for Poisson distribution, and this is our parameter that is a dispersion parameter. The curves looks quite similar to Poisson distribution. Sometimes negative binomial distribution is called Poisson gamma distribution because your rate lambda in Poisson gamma distribution actually follows this gamma distribution. So what you add up is variability in your rate. And this is the plots coming from the summary of 18 studies on 60,000 RNA-seq human samples. They try to plot the real values. This is mean gene expression level on x-axis, and this is a full gene level variance on y-axis, all in logarithmic form. So that plots both lines for Poisson distribution and for negative binomial, and this is, you can clearly see that negative binomial is much better just by eyeballing. Negative binomial is much better for this type of samples. 60,000 samples were generalized. So over here you can see a clear difference between Poisson distribution and negative binomial. This is just a different lambda for negative binomial on the right side. And again, the main difference between them is that in Poisson distribution you have your mean and your variance equal to each other. This is the same parameter as lambda, right? And many people think that it's a cool distribution because it just simplifies many mathematical operations behind Poisson distribution. And in negative binomial, you don't have them equal. You have variability in your lambda parameter. And because your p-value is based on a distribution, your p-value will change if you switch from Poisson distribution to your negative binomial distribution. And let me talk a bit about DSEC2 package, because you will be probably working with it later on in your career life. So what DSEC2 is using, it doesn't use the normalization matrix that I mentioned earlier. It uses something else. It normalizes the gene counts by geometric means. And why is it important to apply different form of normalization? Sometimes you see in sample 1 and in sample 2, the library counts are the same, but let's say that sample 1 is coming from liver and sample 2 is coming from plant tissue. Then you see that the library size might be the same, but these sizes are coming from different genes. And you need some form of normalization to address these counts, and your RPKM normalization won't be able to handle it. I don't have enough time to go into details about DSEC2 normalization, but behind this is a geometric mean normalization log scale. And what is cool about DSEC2, it uses also shrinkage of dispersion per gene. So you can imagine a bowling player. I'm trying to explain again the intuitive understanding of it. Imagine a bowling player, a new player that is coming to a competition. You see a couple of games, and you see that this bowling player generates strikes in 90%. And you know that the actual rate is probably much, much lower, because you know that the distribution of, let's say, inborn rates for the bowling players is around, the mean of this distribution is around 75% of strikes all the time. And let's say that you need to bet for this player, and you need to predict what kind of bets you would like to put on it to get more money out of this bet, right? Would you put a really high bet for the player or not? Probably not, because if you see more games, his distribution will probably be shifted, because you see more samples. Well, not his distribution stays the same, but because you see more samples, there is a high chance he was lucky enough to get this 90% of the strikes in the first games, just because it was a chance, or, yeah, he was lucky. But if you see more sampling, probably you see that this shifts towards 75%, towards the mean distribution value, mean of the distribution for all the players. So whenever you see this 90%, you are suspicious, and you ask to bet. When you're betting, you need to remember about this, and you're saying, okay, probably he will be actually worse in the next games. My bet would be not as high as someone else would bet without knowing statistics. So you bet that this player would be probably around 75%. Your bet will be lower. This is what you actually see in machine learning too, because some noise is involved in your sampling, in your data analysis too. You try to get rid of the extreme values, because you think that these extreme values are actually coming from noise, and you try to shrink those extreme values to the median value. So what DSEC2 does is it's taking into account the mean value that is based on geometric mean, and it's trying to shrink extreme value to this mean value. So the same for the bowling player, because you expect that this will behave slightly worse in the next iteration. And you know that this probably comes from noise. I think those plots are not quite good representation of this shrinkage improvement. But what you see in DSEC, there is so-called MA plot, and you see that it's logarithmic value of fold change, and let's say 0. What you see is a pure spread of those are reads counts, those are counts, normalized counts on x-axis. And you see lots of the spreaded dots over here, and it's actually becoming narrow, more narrow with x. So what you're trying to get rid of, this big spread. Because you can imagine that if you have 2x fold change, in the low counts, if you have just 5 counts for one transcript and 10 counts for the same transcript in sample 1 and sample 2, it's still fold to change, but it's not similar to 1,000 counts versus 2,000 counts. So you might be wrong estimating this fold change for low values, right? Because of just how equipment works, and if we are able to identify those big changes on the lower scale level. So for low expressed genes, it's easier to make mistakes. And it's what DSEC is trying to do is trying to shrink these low counts closer to x. So I have better distribution of these low counts later on for x, and then you might be fooling yourself a bit less. And people try to estimate whether it improves reproducibility, and it does, briefly. So for HR and DSEC, those are two, I think, mostly used packages. And HR, both of them are actually efficient and fast and easy to use. HR provides exact test for comparison between two groups, and it has some extension to robust version against outliers robust. And what is not that good about those two, they don't have random effects included. So if you have like within person variation or between person variation, it doesn't handle it well, both of them. And it cannot actually handle pair treat data. So for pair treat, you need to use something else. And also, because it uses negative binomial, if you have the excess of the zeros, just because, for example, low sequence coverage, low depth, you have so-called zero inflation. So you might get rid of your zeros in your counts on genes because probably it's not coming from real data. Probably it's a result of it's artifact from your equipment. So this is what negative binomial predicts. Amount of zeros located over here, but actually you see in your real data amount of zero is very high. That's why those packages are not, I'm not that good at handling zeros. But we also have shrink bias, which is based on Bayesian statistics, and it's better for small sample sizes because DSEC and HR are applied typically for large to medium to large sample sizes. And shrink bias also handles well sample sizes, and it can deal with also query traits, random effects, and excess of zero. So perfect software piece, right? But because it uses Bayesian statistics, many scientists and many scholars are actually not quite comfortable about Bayesian statistics, right? And probably they don't understand it. So it's more time consuming. This package is actually more time consuming when trying to analyze your RNA-seq. And because we have this familiarity bias, not that many people uses this, right? But it still produces FDR, and with FDR, we're all familiar with, right? So we might want to use to apply this package one day or another. That is it about RNA-seq. Coffee time.
Programs, and they might differ in structure, function, location. It can even be that there's a stop codon in additional exon and then protein is not even built, so RNA is degraded. There are different splicing events. What can occur is basically constitutive splicing, or so this is a normal situation where exons are joined together and intron is removed. But there are also alternative events. So alternative 5' splice site is when part of the exon can be either shorter or longer, and the alternative 3' splice site is the same, but on the other side of the intron. And then skipped exon is when exon is basically removed. And retained intron is that when intron is actually ending up in the final protein, final transcript and then protein. And mutually exclusive exons is when you get one exon out of the two. Now because of this, you're gonna not see any of the references. I cannot move it actually.
Is it stuck now?
Yeah, it's stuck.
Right. If you want to see the references, look through the slides after the lecture.
And then there is splicosome, that's a huge complex that's responsible for removing the intron. And for the sake of time, I will not go into details. A big part of this complex are splicing factor, which are basically proteins responsible for alternative splicing. And now alternative splicing plays a role in the hallmarks of cancer. And so there are different processes that play a role in the cancer development and progression. And then for each process there are different genes that are alternatively spliced in cancer, so their different transcript is expressed in cancer than in normal. One example is BCL2L1, that's alternative 5' splice site, where one form is pro-apoptotic, so the healthy individuals have this one, and the other isoform is anti-apoptotic, and then apoptosis doesn't go on in the cancer cells. And then again, another example is pro or anti-angiogenic isoforms of VEGFI, so either the angiogenesis can take place or not. And then also splicing factors can be aberrated in cancer. So SF3B1 is a splicing factor that's very often mutated, mostly in hematopoietic malignancy, but also in solid tumors, and it plays a role in early splicosome assembly. What it's actually doing is sequence-independent free splice site recognition, so it has more of a global role, while SRSF1 is another splicing factor that does sequence-specific splice site recognition, and it's often differentially expressed in different tumors, also in colorectal adenocarcinoma. Oh yeah, and then someone even wrote a review on the splicing factor, how it affects different hallmarks of cancer. So in the translational research, you can work with alternative splicing in multiple manners. You can target splicing factors or mutated splicing factors with inhibitory drugs. You can target aberrantly spliced mRNA or target the protein product created by alternative splicing. And what we do is actually identify aberrantly spliced proteins as biomarkers for early detection of colorectal cancer. So very quickly, colorectal cancer has a high cure rate when it's diagnosed early, so at stage 1. While it progresses, the 5-year survival rate basically drops down, which is why early detection is important. And in the Netherlands, there is population-wide screening for colorectal cancer that bases on fecal immunochemical tests. So people from 55 to 75-year-old get this tube, and they have to take a sample of their stool. And when there is protein hemoglobin above the certain threshold in their stool, they are FIT positive, and they're referred to further colonoscopy to confirm if they have cancer or not. And while FIT performance is quite good, it can be still improved, so there's still over 20% of people with cancer that are not detected with this method. And it also concerns cancer precursor lesions that FIT has lower sensitivity. So there's clinical need for a novel biomarker, and we will would like to look at alternatively spliced mRNA as a source of potential biomarkers for colorectal cancer screening. Now, if you could think with me on this Monday morning, why would alternative splicing be interesting for this question? Yeah?
You could find junction proteins that are not expressed in healthy individuals, but only in cancerous patients.
Exactly. So a lot of these alternative spliced mRNA variants are translated into protein isoforms, and then the proteins really differ in structure. So you could have more qualitative measure who has cancer or not than the quantitative in terms of the level of protein hemoglobin that's in the current test. So that was our idea for this project. So today I'll talk about how we design an approach to identify these two more specific protein variants, and that's in the manuscript that hopefully you'll read. So we will use mass spectrometry to identify these protein isoforms. This is a typical mass spectrometry experiment. So there's experimental part where you cut the protein into peptides, usually with tryptic digestion, so with an enzyme that cuts a specific locus, so lysine and arginine, and then you obtain mass spectra from the mass spectrometer. And how do you match this spectra to the original peptide? It's based on the in silico analysis, where you have protein sequence database. You cut your proteins into the tryptic peptides, so you always know where to cut based on their amino acid composition. And you build theoretical MS pattern of the peptides based on the amino acid sequence, and then by matching these you can identify peptides and then later on proteins. So the big problem with this approach is that you're limited by this protein database, so we will use RNA sequencing to look for alternatively spliced variants, and will basically enrich this protein database with our findings from the RNA-seq. So this is the whole project overview, where we start with colorectal cancer cell lines when we really need to have positive control that our pipeline works. So we downloaded two splicing factors I mentioned before, and we have control that's non-targeting, and we established splicing controls to see if our results are correct, and then we do RNA sequence proteomics, based on which we design proteogenomic pipeline that will be applied later on the tissue samples. And after we have a candidate biomarker, we will move to an antibody-based assay to detect these protein isoforms in stool and in FIT samples. So today I'll talk about the design of the pipeline, so we'll stick to the colorectal cancer cell lines. So this is typical RNA library prep for Illumina. You probably saw this before, but this is where poly-A RNA is captured, so it's already after the splicing, and then RNA is fragmented into small pieces, and these vary depending on the sequencing run you go for, but these are basically quite short, up to 250 base pairs, while the distribution of the exon length for human is actually different, so majority of the exons will be within your read length, but there are also a lot of exons that are longer. And this makes the analysis of alternative splicing quite complex, as you have to look at the specific exon-exon junction, so you would preferably have reads that really span your exons. But there are two different methods how people approach this. One was transcriptome assembly, where you obtain the whole isoforms, and this can be done reference-based. That's done for the human because we know the sequence of human genome, or de novo when you have unannotated species. or you can look at the individual splicing events. So the problem with transcriptome assembly is that it's actually very complex, and there is plenty of tools that approach this issue. And basically they work as follows. So you have your reads map to the genome, and you build a graph representing where a node is representing a read, and edge is when two reads have overlapping sequence. And then you traverse this graph to assemble the splice variants, and that usually takes a lot of assumptions. For example, people take the lowest number of isoforms that can explain this graph to get your assembled isoforms at the end, and then you can move on to isoform quantification. So the problem of transcriptome assembly is sensitive to mapping software, a lot of assumptions, how to traverse this graph, how to quantify transcripts if if reads map to multiple isoforms, and little concordance between different transcriptome assembly tools. But the big asset is that you have the whole, the whole isoform. And then the other thing is differential splicing, where you look at individual splicing events that I mentioned before, skipped exon and things like this, where you look at the reads only in the region you're analyzing. So basically you look at these exon spanning reads, and you quantify the inclusion spanning reads, so the ones supporting inclusion of this exon, and exclusion spanning reads, so the reads supporting exclusion of this exon, and you can calculate the psi values. It's percent spliced in, so it's also called inclusion level, where basically you calculate the percentage of your reads supporting inclusion transcript, and then percentage of your reads supporting exclusion transcript, and you do it for both, for two conditions, very often to do it in a differential setting. So you compare this inclusion level in sample 2 to inclusion level of sample 1, and you get delta psi. And then when you plot the distribution of these psi values, usually they correlate for non-differential events, while the red and blue are higher expressed in one of your conditions. So you can extract those also with statistical testing to identify differential splicing events. And authors of the tools that provide this analysis, they claim you can even validate 85% of the computational results with experimental methods, so with quantitative real-time PCR. So now we have these two methods, and well, obviously you probably know I went for the second one, as the title of the talk is differential splicing analysis, but it would be great if you could think with me, why would we go for the right one and not for the left one, and what are the advantages and disadvantages of both.
Don't be shy.
So maybe a tip. Ultimately, we would like to improve clinical practice, right? So we would like to have a method that wouldn't give us a lot of false positives. Not to detect something that basically is an artifact of our computational analysis. We went for this method because of the very successful experimental validation of the results. So we basically want to have events that we really believe are true. While here we could do, we could run a lot of transcriptome assembly tools and then look for overlap to be confident of our results. But there's another thing in our project, and that's proteomics. And as I mentioned before, you get peptides from your proteomics experiment. So it's also a local confirmation of your alternative splicing. So you cannot really confirm with peptide analysis the whole isoform because these peptides are not long enough. And then there's a trick in this question, and that's basically novel methodologies of sequencing. So there is not only Illumina available on the market, but also long read sequencing. So what we actually did is also used PacBio IsoSeq methodology, where you can sequence full-length transcripts. What happens is poly A reads are extracted as for Illumina, but then adapters are added straight to the whole reads, and then there is a size partitioning required for this methodology. And basically for each transcript you sequence kind of the whole transcript in such a manner, and then assemble the most frequently occurring sequence into your isoform. And they also have a special computational pipeline to do this analysis. Big advantage is you really get full-length isoforms, so you're really sure what's in your sample. Disadvantages are quantitative analysis is impossible due to this size fractionation, so to enrich for each size of the transcript, and quite high costs compared to Illumina. So this is our experimental design. We do RNA sequencing with Illumina for shorter reads and proteomics for all our conditions, and then for one condition with down modulation of SF3B1. And the control, we do also PacBio IsoSeq to identify full-length transcripts. And this is how we design our proteogenomic pipeline. So our input is short RNA-seq reads and mass spectra. We do the standard processing of RNA-seq, I hope you know it, so quality and adapter trimming, reads mapping, and then we do differential splicing analysis to obtain this differential splice variance on RNA level. And for this we need reference annotation, so the tool really needs to know what are the coordinates of exons and introns to do this analysis. And then we do free frame translation, so completely theoretical what amino acids we would get from this nucleotide sequence. And we get a database of potential protein variants that we enrich with the human protein database from UniProt. And then we use this database for mass spectra identification to extract our variant peptides, so isoform-specific peptides, and then we do differential peptide analysis to confirm the quantitative difference from RNA level on protein level. And then how do we use the IsoSeq data? Oh, I'll go to it later apparently. Okay, so these are the results. So we get an event, exon skipping, for which there is differential inclusion and exclusion expression between our two conditions, and basically exclusion level, just a reminder, is number of exclusion spanning grids divided by number of exclusion and inclusion spanning grids. So for 5 different splicing events, we obtained number of these events for each condition, so the modulation of SF3B1, which is the not sequence-specific splicing factor, and down modulation of SI-SRSF1, which is performing more has more targeted role in the cell. So obviously for this one we obtained more events. And then this is an example, so a skipped exon 9 of the gene OSBPL3, and the exclusion level differential between our down-modulated condition versus control. And we had confirmation of this difference with real-time qPCR. So we did a number of these validation experiments with real-time PCR for our interesting candidates, and then we moved on to protein analysis. So maybe also a question to you: how would you, based on the events that you obtained from RNA sequencing, how would you confirm an event is translated to a protein? So what evidence do you need from the proteomics experiment to do that? Mm-hmm.
You need to find peptides from that region, right?
Yeah, exactly. So you need isoform-specific peptides. So this is an example of exclusion-specific peptide that will not map to this sequence because of the red exon. Inclusion-specific peptide that will not map to this sequence because of this Y. And then inclusion-specific peptide on target that maps fully to the region that's spliced in or out. And that we can identify for different kind of splicing events. So this is an example of retained intron, where now the purple part is the intron, not an exon. And you can have inclusion-specific spanning peptide that spans exon-intron junction that will not map to the exclusion variant. So we identified a number of this isoform-specific peptide, mostly peptide on target. That's because some of the exons that are spliced out are really large, so then a lot of peptides can map there. While for split and spanning peptides, you really have to have specific sequence to have the right composition of arginine and lysine. It's where trypsin cuts to obtain the peptide in the proteomics. And then this is the number of events on RNA level. Well, this is what we saw on protein level. So for some of the events, we get, let's say, 40%, while for others, 20% with peptide confirmation. And this is example of our OSB-PL3 transcript, where we have either inclusion of the exon or exclusion. This is screenshot from IGV genome browser. So basically blocks are exons and the line is intron with the direction of how the transcript is transcribed. And these are just a reminder, quantitative differences that we saw between the two conditions based on RNA-seq. And these are our isoform-specific peptides that we identified. So peptide spanning exon-exon junction characteristic for inclusion variant, that's again in here. These are the same peptide, they only differ in one amino acid. It's sometimes trypsin makes a mistake, and that's what you get, so you should add this up. And then you have one exclusion-specific peptide that maps partly to this exon and partly here. And then when you do quantitative analysis, of course, these peptides intensities are very specific to the amino acid sequence, so you cannot compare different intensities of different peptides, but you cannot compare intensities of the same peptides between different conditions. So you can see inclusion variant is higher expressed in the non-targeting, that's what we saw here, and exclusion, so this exclusion level is higher expressed in the SF21. So when we do it globally, we obtained around 65-64% of the peptides concordant on quantitative level with the RNA results. And here comes another question, why not all? So this is a comparison of the quantitative analysis, RNA-seq versus proteomics. So let's say in RNA-seq, we know exclusion variant is higher expressed in your condition, SA31, whatever, while inclusion is higher in the non-targeting. So then when something is consistent, it means on protein level, we saw the same quantitative differences, while when something is opposite, it means on protein level we don't see this, or it's not significant, and things like this.
Well, there's regulation in translation, right? So if
Yeah.
you have a specific number of mRNAs, it doesn't mean they're all translated equally
Yeah.
or at all. That number.
Exactly. So transcripts can be degraded, there can be different kinetics, so something is transcribed but it takes longer to translate it into protein, or protein is degraded, and things like this. Or actually active, how do you call it, suppression of the expression of the certain protein isoform. That could also be the depth of our experiment, so the technical issues, differences between RNA-seq and proteomics, and so on. Okay. So what did we do with our full-length transcript? So far I didn't talk about this. So this is what I talked about, this was our pipeline. And now what we do is we take our full-length transcripts and we use them instead of reference annotation. So we provide our pipeline with the exon intron coordinates from the experiment and not from the basically available databases for human genome. So in both approaches we still use the short RNA-seq reads of Illumina to do quantitative analysis. So this is the comparison that we obtained, PacBio full-length transcript versus reference annotation. We got more events with PacBio, and while enrichment for the skipped exon and mutually exclusive exons was not that significant. For alternative 5' and 3' splice site and retained introns, we really got more events with the PacBio isoseq. Well, we also have event specific for reference annotation, and it might be due to the sequencing depth of our PacBio experiment that we didn't sequence all of the transcripts. So these are our novel unannotated events that are in particular interesting for these down modulations of the splicing factors. And this is the number of isoform specific peptide, again, more for the PacBio data analysis approach than for the reference annotation. And this is an example. So what you can see, this is the reference genome. So these are the exons, introns, exons, and this is the coverage of your reads. This is non-targeting control versus your condition, so SR3D1. So you can already see the difference between the two plots. There's way more reads mapping to the intron. But somehow with our reference annotation approach, this was not identified. Well, if you give the PacBio transcript to the protogenomic pipeline to splicify, it identifies basically retained intron, so this is the inclusion variant of the intron, exclusion variant of the intron, and then we also identified isoform-specific peptides. This is the same peptide with two more amino acids at the start, but basically both peptides are spanning exon-intron junction, so really a confirmation that this intron is translated into the protein. And this is another example. Again, exons here, the intron, and you can see there are some reads mapping here. While RevSec annotation doesn't really note that there can be an exon in here. But the PacBio transcripts did identify it. Interestingly, they claim the exon finishes up in here, not so far. And then there's difference in coverage between these two conditions. So we identified alternative 3' splice site for this transcript where this exon is either longer or shorter. And then we have isoform-specific peptides for both transcripts. So really, you can see that they both map in here. So the beginning sequence is the same up till B, I think. and then the rest maps to this region. So the rest of the peptide sequence differs. So again, confirmation on protein level of both isoforms. So this brings me to the conclusion. We built Splicify pipeline, that's protogenomic pipeline combining RNA-seq and proteomics data for differential splice variant identification, that's differential between two conditions. We used it in the same cancer cell line down modulation of a splicing factor versus control to really see splicing changes, but then late in the project to apply it to cancer tissues versus healthy controls. We showed there's confirmation of these splice variants with two different techniques. And we identified novel splicing events with PacBio IsoSeq, which was super exciting that we got confirmation on the protein level. But another lesson we can learn is that reference annotation of the human genome can be still improved, that they don't know about all the exon intron coordinates. There were interesting biological findings for to study further relationship between splicing factors and identify isoforms. For example, we identify MKI67 short and long isoforms that were shown to play a role in cell differentiation, and how do these splicing factors might regulate cell proliferation through alternative splicing of this isoform. And then, of course, there are limitations in this approach that we discussed briefly. So, biological, not all RNA transcripts are translated into proteins, so you cannot identify everything in your protein experiment. And then RNA and protein expression are not always concordant. There are post-translational modifications in other processes to basically make the cell live. And then a lot of technical issues. So tryptic digestion is very specific, so it cuts at two specific amino acids, arginine and lysine, and they're not always in the vicinity of the exon-exon junctions. So sometimes you just cannot identify the peptide in this region with your methodology. And also limited depth of mass spectrometry experiments. We really operate on the peptide level, while the coverage of the peptide information is not as high as the read coverage for RNA sequencing. And then ongoing work is we analyzed human tissue samples from normal adenomas in cancers and identified a number of potential candidate biomarkers. And now we've built antibodies for our best candidate to test the quantitative differences between different isomers. And with that, I would like to thank everyone, both groups I work with and collaborators from the PacBio that did the PacBio IsoSeq, and HAVF for funding, and serve Sarah for the use of their computational hardware.
All right. Thank you, Gosia. So she has pestered you with several difficult questions throughout the presentation. In view of time, there is opportunity for you to return the favor. Any questions for Gosia?
Yeah?
Sorry. Illumina uses 125, baseline is 125, right? But the average human exome length is 200, right? Would you think that the technique you're using would improve if you had like longer reads? Because then you probably, because the regions where you're interested in are they're probably at higher scale.
Definitely. So that's a very good point. So obviously, when you design your sequencing experiments, it's a balance between the finances and what you want to achieve. But basically, the longer your read is, the higher the chances that it's going to span, exon-exon junction. So definitely, you want to go as long as possible.
Did you guys try to use longer reads or is it?
No, so we did this by bioisoseq, so that's the longest read possible. And I mean, it's not that easy to get everything you want, right? So it's not a priority to resequence the same samples with the same method, but slightly longer reads to compare longer versus shorter. Could be a project if someone is rich. No, I mean it's just not a priority. You would rather sequence more tissue samples later in the project than do this technical exercise.
I understand. Thank you.
No one? Alright, then let's then go see again.